Got quite a few of you on the homework assignment that was due last week Friday. We have a dominant trait polydactyly, individual 2-4, has 6 fingers on the right hand, while individual 4-3 has an extra toe on each foot. Individual 3-2 has 10 fingers and 10 toes. And the question is, these individuals show a range of incomplete penetrance. That is false. There is usually no such thing as a range of incomplete penetrance. Penetrance, remember, is just basically the probability that an individual with the genotype has the trait. It's very, very much a binary thing. We're here talking about individuals with either extra digits on toes, or in some cases on the hand, and different numbers on the hand, and that is a range of expressivity. So what this would really be reflecting is expressivity, not penetrance, which is more of a binary, are there extra digits and toes, as opposed to where and how many, which is what we're seeing here in this question. Yes?
So I was confused on that one because it mentioned individual 1, 3, 2.
Yeah.
And that one, like they're heterozygous and they don't show.
Yeah, that one individual is incomplete penetrance, but we're talking about the individuals collectively showing a range of phenotypes, including none. But I see where that could be confusing. The other one, which I wrote on for the previous class and is still written on, but I'll just keep it. We have a couple who are carriers for the sickle cell allele. They have 5 children. Tells you a lot about their children, but it doesn't really matter what they did. They're only asking about a 6th child. And remember, each child is an independent event. And so in the 6th child, what's the probability that a phenotypically normal child is a carrier? So if you were to do the Punnett square, many of you probably put in 50% because the probability of having a carrier is 50%. But recall, they don't have sickle cell. And we know that because they're phenotypically normal. So what is the probability that a phenotypically normal child is a carrier? It's the 2 out of the 3 remaining squares of the Punnett square. Very similar to the question from a pedigree a week ago, where the question gives you more information than just a straight Punnett square. Any questions on those? Yes. Yeah. So incomplete penetrance is just, so the phenotype of polydactyly is extra digits and toes, however defined. And so if you have incomplete penetrance, that means that you have the dominant allele, but you don't have any phenotype at all. That's incomplete penetrance. The disease allele or whatever doesn't penetrate through to phenotype. It's masked in some way. So expressivity is again more about how many fingers and toes, where are they, the variation in the trait itself. Not really, because they have the trait. So they would be just polydactylous. I guess they would have a penetrant allele. You could think of it, just not without the incomplete part. Okay. Let's move on then to today's discussion, soon to be your favorite discussion, linkage. So we'll talk about what it is, how it's associated with recombination. We will use a chi-squared test to see if we have support for linkage happening. We will determine gene order as well as the rough statistical distance between genes, all today, which will be a lot of fun. The goal that I have for you is that you'll be able to understand linkage and its implication for non-Mendelian inheritance patterns. We will talk about a lot of non-Mendelian inheritance patterns shortly after our exam. And one of them that we will talk about today is linkage. We will test, I want you to be able to test for violation of independent assortment or non-linkage using chi-squared tests. And then finally, to analyze results from linkage mapping experiments to determine if genes are linked, how far apart they are in map distance, and the relative order of genes on a chromosome, all from counting progeny numbers. So just by doing a specific cross and counting the numbers of different phenotype combinations that you have, you can determine all of that, what chromosome genes are on, how far away they are from one another, and their relative order on a chromosome, which is a very cool and powerful old school technique here in linkage mapping. So let's start with linkage and what it is. So the basic idea of linkage is actually quite simple. It's that genes are close together on a chromosome and that those genes are transmitted together. All right, so we're talking about genes physically close to one another on a chromosome. Alleles of those genes will likely be inherited in tandem with one another. And the reason that they'll be inherited in tandem with one another is due to the rarity of recombination. We will actually draw this out in a minute. What this means is alleles, or variants of genes, of certain traits may cause a co-occurrence of these phenotypes. Which violates the law of independent assortment. So if you recall from Mendel's experiments, you would take a purple flowered pea and he'd cross it, that was tall, and he'd cross it with a short plant that had white flowers, and he would get a mixture of all of those different phenotype combinations, right? He would get short plants with purple flowers and tall plants with white flowers and all possible phenotype combinations. If the genes, however, are closely linked together on a chromosome, if they are close together, it would be a situation where certain traits would often co-occur with each other. So if the mutation that caused white flowers was really close to the mutation that caused short plants, you would always see short white plants together, and you would rarely see a situation where you had tall plants with white flowers, or et cetera. You wouldn't see them mixing. We'll hopefully illustrate that throughout the course, or throughout the class today, and that'll make a little bit more sense. So when we say linked, again, we mean very simply, these are our chromosomes here. We have two genes, and they're near each other on a chromosome. Each other. All right, these are linked. They are likely to be transmitted together during meiosis. However, if you have genes on chromosomes, and maybe they're on two different chromosomes, or maybe they're far apart on the same chromosome, these are unlinked. And they will behave just like you would expect in Mendel's law of independent assortment. So these are on different chromosomes or far away from each other, and they are unlinked, which means they are more likely, at least in the case of far away, to have a crossover event or recombination. So recombination is rare. The odds that you have a recombination event that happens between gene 1 and 2 is pretty low. But if they're far away from one another, it's an increased chance you could have a recombination event anywhere in that space. And so you're more likely to separate an allele of gene 1 from an allele of gene 2. We'll draw that out in 1 second. But again, when you're thinking linkage, that just means they're physically near each other on a chromosome. So it's just pretty straightforward at its core. So ultimately, this reflects recombination. And recombination is what breaks linkage. All right. So if you recall, this all happens in prophase 1 of meiosis, where we had, I think in one example, we had the blue chromosome of, the one from dad, let's say, and we've got the red chromosome from mom. They synapse. That synaptonemal complex forms, a chiasma forms, and they cross over, giving us something that was a mixture of the red and blue chromosomes. So one blue chromosome with a little bit of red, and one red chromosome with a little bit of blue. That's what's happening in recombination. So it turns out you can have what are called single crossovers, where, as we've drawn here, a single piece of chromosome is swapped between two chromosomes. It can also happen twice and give us double crossovers. Let's start with a single crossover. So let's draw a chromosome, and let's give it some dominant and recessive genes. So we've got the red and the blue chromosome. Here we're laying it sideways for ease of drawing. We'll have a dominant A allele, a dominant B allele, and a dominant C allele. And those will be on the blue chromosome. The red will have a recessive version of each of these alleles. If there's a recombination event, say between A and B, this little chiasma forms, and it's like we take a scissors and cut these and then swap the red and the blue pieces, such that we have a chromosome now that starts with the dominant or the blue chromosome and then switches over after the crossover to be a red chromosome. We have a chromosome that starts red and then switches over to end in blue, giving us something that is a dominant A allele, but now, rather than being next to the dominant B and C from the original blue chromosome, this A allele is next to a recessive B and a recessive C from the red chromosome, while the other chromosome has now the recessive A allele with the dominant B and the dominant C allele. Right, so a single crossover event takes an A and a B allele that were dominant and were always associated with one another. Now you've mixed and matched the dominant and the recessive alleles from those two parental chromosomes. Does that make sense? So this can happen once. It can happen between A and B. It can happen between A and C, right? You can also have what is called a double crossover. Which, as the name might suggest, involves two crossover events. So let's draw that out. We've got the dominant A, dominant B, the dominant C. We have the recessive A, the recessive B, and the recessive C. We previously just had one recombination event here between A and B. Now we also have a second one between B and C. That gives us a very interesting banding pattern. Like so. So the result of this cross is we have a chromosome just like here, we've cut and pasted the red up here, right? So we've got this recessive B now with the A. Now there was a second recombination event, though, that swapped this back over and put the dominant C back in the same chromosome with the dominant A. So that looks something like this, and the other chromosome is just the opposite of that. Right, so we cut it, we swapped one side, and then we did it again and swapped the other side back over, right? It wasn't at the exact same spot. So you have this banding pattern where the gene in the middle is the only one that's different between the three genes shown here. Does that make sense? Thing to keep in mind, double crossovers, which I will abbreviate DCO, that is a rare thing. So a single crossover is actually itself pretty rare. A double crossover, even more rare. And I'm not going to draw them out, but the textbook does illustrate that there are many ways to get double crossovers involving all four of the sister chromatids that are present in the homologous pair up here, right? So these guys can mix and match in multiple different ways to give you a double recombinant, but I won't go into that because I don't think it's all that relevant to the types of questions we will be going through in class. The recombinants these are going to be any phenotype combinations not seen in the parents. These will also be called non-parentals. So in this case, our parents had a combination of whatever the phenotype is of the dominant A, the dominant B, and the dominant C. Right? This individual has the dominant A, but now it's with the recessive B and the recessive C. That would be a new combination of alleles and a new suite of phenotypes potentially not seen in the parents. These would be called non-parentals.
So basically, whenever a crossover occurs, the occurs will turn on.
Correct. Yep. Any crossover will generate non-parentals. A new chromosome that was not the same chromosome seen in any of the parents. All right, we'll draw this out in a little bit of detail in an example in a minute. Before we do that, we should discuss the ultimate application of all of this, which is linkage mapping. And the basic idea of linkage mapping is that you can take traits, such as whatever shiny new mutant you happen to have, and they can be mapped to a specific chromosome, or, as the more historical phrase, to a linkage group. So as a result of a lot of work over many years by a number of labs, for example, we have linkage maps that look a little bit something like this. Imagine we have 3 chromosomes, or linkage groups. We'll go chromosome 1, 2, and 3. And in Drosophila, for example, we might know that on chromosome 1, we have a allele that causes a yellow body phenotype. We might also have an allele that causes white eyes. And we have an allele also on this chromosome that causes dumpy wings. A little insulting, but... Then on chromosome 2, we know there's an allele that causes sepia eyes. And there is an allele that causes short bristles. Chromosome names made up here. I don't actually know that these all go together. And on chromosome 3, we might have an allele that causes long bristles. Okay. This is essentially a linkage map. Remember, chromosome equals linkage group. So long before we had genome sequences and that we knew all the sequences of all the chromosomes and where all the genes were, you might have isolated a new mutant. Let's call it pretty wings. okay, instead of dumpy wings. And you might want to know where in the genome the pretty wings allele is, right? Through a series of strategic crosses, which we will go through at depth in a minute, you would be able to say, well, it seems to be linked to the sepia I genes, because whenever we cross it to the sepia I mutant, there's a biased pattern of trait distributions, right? And we'd be able to say it's not only between sepia I's, it is roughly a certain genetic distance from sepia I's, and it's between that and short bristles, right? So you can place, just by counting phenotypes, roughly where in the genome a gene is, how far away it is from other genes, and the relative order of those genes on the chromosome. That is all through linkage mapping. And so before we'd had genome sequencing, you would be working in a lab and you would be systematically crossing your mutant to a lot of other mutants to figure out where in the genome that gene is, and ultimately try and figure out what the gene is in terms of gene sequence that was mutated. You can use, therefore, something called a map cross to find where a gene of interest is located. All right. Now, if you want to know how far away genes are from one another, you can calculate what is called a map distance. The map distance is the number of recombinant offspring. So these are the non-parental phenotype combinations. And you can divide that by all of the offspring you're counting and multiply by 100. The units here are map units, very clever. Okay, also abbreviated MU, or sometimes referred to as centimorgans, or CM. Effectively, one map unit, you can kind of solve this as basically a percentage, right? One map unit is a 1% recombination frequency. Meaning there's a 1% chance of having a recombination event between those two genes. If those genes are further apart, let's say they're 20 centimorgans apart, that means there's a 20% chance that there's a recombination event between those two genes. And the further away you get, the higher that percentage likelihood ultimately gets. Now, importantly, this is not a physical distance. It correlates roughly to a physical distance. But if you're one map unit away, that does not mean you're 100 base pairs away in DNA sequence. It does not mean you're 100 nanometers away in physical distance. It's just a probability of recombination, which does happen to correlate with the distance between those genes. Question?
So the lower the recombination frequency, the more likely it is that the genes are linked?
Yes. Yep. So the lower this number, the closer the genes are, the more linked they are. Centimorgans. Does that make some sense? We're going to go through an example now where we try and sketch out the whole process. All right, this will involve, in every good map cross, a test cross. A test cross is used for mapping gene distances and ultimately in determining linkage. The test cross is composed of one heterozygous individual for all genes that we are discussing, in this case usually 2 or 3 genes of interest, and they will be crossed to one individual that is homozygous recessive for all genes of interest. This is the essence of the map cross. We'll talk about why the homozygous recessive thing is important in two different places, actually. So let's go through an example. We're going to start just one cross before the test cross. And let's draw out an example of a mutant with two traits. So let's do a fish example. Got to give people what they want. More fish. Okay, let's give the long fins on this fish. Look at that. And that fish will also have stripes. Give them some eyes here. Look at that. All right, L will be a long fin. And S will be stripes. And their genotype will be homozygous dominant for each. And they will be crossed to an original parent that has short fins and spots. All right, so we've got a fish here, short fins and spots. They will be homozygous recessive for both. This is not yet the test cross. Now, if we were to draw out these genes on a chromosome, we would have something that looks like this. So let's say they are on the same chromosome. This individual will have a purple chromosome. That purple chromosome will have a dominant L allele and a dominant S allele. And remember, they are homozygous. The other individual parent will have a yellowish chromosome here. And they will have the recessive alleles here. Okay, so if we cross these two fish together, we will end up with getting one chromosome from this parent and one chromosome from this parent, right? So you'll have one purple chromosome and you will have one yellow chromosome, and they'll be present in the same individual fish, and they will be heterozygous. So this individual will have the dominant S allele and the dominant L allele and the recessive S and the recessive L, their genotype being something like this. Okay, so now they are heterozygous. This one individual has both types of chromosome. They will then be crossed to the test cross that is homozygous recessive for both alleles. So they're L and S, L and S. This here is the test cross. And the beauty of the test cross is, one, when it comes time to do chi-squared tests, which we will do in a minute, it really simplifies your analysis greatly. The other is that it really lets you see recessive phenotypes more easily. And in particular, it allows you to only focus, so it lets you only see recombination events from the heterozygous parent. Ooh. All right, so recombination is rare, remember? We want to see how many recombination events we count between the L and the S. This parent, there will be a recombination event, but it's just recessive to recessive. It'll be invisible. You'll never see it. So anytime that you have recombination events, you know it was from these two chromosomes. And that'll greatly simplify our math. Plus, because recombination may or may not be occurring, you need a recessive background to compare it to so that you can see the recessive phenotypes when they first emerge. So what does that look like then? We can have, as the product of this, an individual that has the pink purple chromosome with no recombination, and then the test cross chromosome, right? So their genotypes would be dominant L, dominant S, recessive L, recessive S. this individual would have long fins and stripes, which is very similar to the parent that we originally had up here. Remember this, that's basically this parent. We could have a chromosome that has no recombination, but you happen to inherit the recessive chromosome and the test cross chromosome. So this individual then is homozygous recessive for both of the alleles. This is an individual that has short fins and stripes. So these are the exact same phenotype combinations we had in our original parents up here, right? So these are non-recombinant. Also called the parental dye types.
What did the second one have spots instead of stripes?
You are absolutely correct. That is supposed to be spots. Yes, thank you. Right, so these are the combinations that we saw in one parent, combination that we saw in the other parent. But if recombination occurred and we start mixing and matching the pink and the yellow chromosome, we would see something a little bit more like this, where we'd have mostly a pink chromosome with just a little bit of yellow, and mostly a yellow chromosome with just a little bit of pink. And in each case, you're also taking along for the ride that test cross chromosome that's recessive for all traits, right? So we've got the L and the S, L and the S. But here we'll have the dominant L mixed with the recessive S, and we'll have the recessive L mixed with the dominant S, giving us, in one case, an individual that is long fins and spots. So that combination didn't exist before in the original parents. And an individual that is short fins with stripes. Okay. These two groupings are recombinant, right? We've recombined the traits of the fin length and the stripe patterning. In addition to recombinant, they're also called non-parental dye types. So if we did this experiment, we can have two phenotype classifications that are the parental, two that are the non-parental, and we can count how many of each we actually have. And if we do that, let's say we count the number of offspring and find we have 537 long fin with stripes. We have 542 short fins with spots. We have 76 long fins with spots, and we have 75 short fins with stripes, giving us a map distance between long fin and stripes of 76 plus 75. So we're counting the recombinants, and then we're going to divide that by the total number, 537 plus 542 plus 76 plus 75. And then we're multiplying that by 100. And that'll give us 12.3 map units. So these genes are 12.3 map units apart. There is a 12.3% chance that in any given meiosis, we have a recombination that separates the L allele and the S. How do we feel about that? Good? Why don't we try our luck at a point solutions attendance question. This will be session ID 119114. 119114. Genes for body color, black being dominant to yellow, and wing shape, straight dominant to curved, are located on the same chromosome in flies. A fly that is homozygous B recessive, so yellow fly with straight wings are crossed to black fly with curved wings. Then we do a test cross, and get the following phenotypic data out. What is the map distance between B and C? Do another 40 seconds. All right, if you could put in an answer, please. I would like to call it. All right. There's one of, all right, good. Let's see. Yep, overwhelming support for A, which is the correct answer. A couple things I think worth discussing here. In the example I gave you with zebrafish, we had a different combination of, all the dominant alleles were in one parent, all the recessive alleles were in another parent. That's not always the case, right? Here we have a situation where you have a mix of dominant and recessive within the parents. There's a couple ways to approach a question like this. One is to say that we have in one parent the recessive B and the dominant C. So anytime you have the dominant B with the recessive C, anytime you have the recessive B and the recessive B together, you have recombination, right? So you have dominant B with recessive B with dominant C. Anytime you have two recessives or two dominants together, like you see here, you have a recombinant. And so those will be what you add together to find the map distance between those genes. The other thing to remind yourself is that recombination is rare, and you can identify here the parents, the parental types, both by the question. We are recognizing that black was with curved and yellow was with straight in the parents, but also by the numbers of progeny, right? You just have a lot of these. You need relatively few of those. And so with barely glancing at the question, you could probably add 17 and 12 together and divide by the total and get the distance between those genes, just vibing off the phenotype abundance alone. So two ways to think about it. I would always go back and make sure that you have a new combination of alleles, but that is one way to do it relatively quickly in a pinch. So let's go ahead and end that. So let's say you have some data and you want to know, are my genes linked or are they unlinked? And so let's briefly discuss testing linkage. We can test linkage with our good friend, the chi-squared test. Where, if you recall, we take the observed numbers minus the expected, square that, and divide it by the expected, and we sum that for all of the phenotype categories. The null hypothesis for a linkage-based chi-squared test is basically the same as any null hypothesis in at least this portion of the course, that there is no linkage, and that you would have basically Mendelian ratios. Right, you're talking about independent assortment here. You would assume these genes are not linked going into the cross. So if you recall, our test cross was a heterozygous individual by a homozygous recessive. And one place that really illustrates the value of the test cross is in the progeny numbers. That's because if you were doing the Punnett square for this specific cross, you only have to worry about this individual giving you a recessive L and a recessive S. So your Punnett square is basically a one-column Punnett square, since that's the only thing they can give you. And then the other option are all the different combinations of alleles you can get from the other parent. So you would expect an individual that is like that. You could expect the dominant L with the recessive S. You would expect the recessive L with the dominant S, and you would expect recessive of both alleles. And you would expect them in equal numbers according to this table. So you would expect 1/4 longfin fish with stripes. These are, of course, if they are unlinked. You would expect 1/4 longfin fish with spots. You would expect 1/4 short fin fish with stripes. And you would expect 1/4 short fin fish with spots. So if you were to sum up what we tallied on the previous slide, we had counted 1,230 fish. It's 1,230. These are the total counted on the previous slide. This is our expected ratio. And that would give us the expectation that if these genes were completely unlinked, we would have 307.5 of each category of fish. All right, these are our expected progeny. So we can take this to a chi-squared test now that we have our observed and our expected to calculate a chi-squared value of goodness of fit. So if we were to do so, we had previously mentioned on the other slide that we had 537 individuals that were long-finned with stripes. We will subtract from that the 307.5 that we expected and divide by the 307.5. We would add to that the 542 short fin fish with spots that we had counted over the 307.5 that we expected. We had counted 76. I can't remember which one it was. We'll say a longfin with spots. And we expected 307.5 of those longfin with spots. And we had counted 75, I think it was, of the short fin with stripes, and we expected 307.5 of those. Okay. That equals basically 171 plus 178 plus 174 plus 175, or a chi-squared value of 689, which is quite a large chi-squared value. I'll give you a minute to sketch that out. So we can then take that very, very large chi-squared value and we can bring it to our trusty chi-squared table. How many degrees of freedom are we going to have? 3. That's right. We have 4 expected phenotype categories. And degrees of freedom are those numbers of categories minus 1. So we have 3 degrees of freedom. Waiting for a few remaining pen strokes here. Okay, so if we go to our table, we have 3 degrees of freedom. So we only really worry about this row. Our chi-squared value was 698. So we then say at a p-value threshold of 0.05, is our chi-squared value greater than 7.8? And it is, so it's much greater than 7.815. It's also much greater than 0.01 if that was our threshold for statistical significance. And so we can conclude that our data does not fit the null hypothesis, right? So something else is happening here that isn't independent assortment of these genes and is likely, in this case, linkage. Does that make sense? All right, one more thing to discuss, which is gene order on a chromosome. We can not only figure out roughly, in at least a statistical way, how close together our genes are, we can determine the relative order of 3 or more genes on a chromosome. And we do this through what is called a 3-factor cross. So let's do another example where we draw out some phenotypes. So let's stick with our fish here. We've got long fins with, here, look at that, nice stripes. And then let's give it another phenotype. In this case, we'll give it long barbels. Okay, so barbels are like catfish whiskers, basically, right? Barbels are covered in taste buds, allowing fish to taste stuff in the mud and whatever. Would you want barbels, a bunch of taste buds hanging off your face? I feel like I wouldn't, no. So we've got long fin. We have stripes. And we have really long barbels. Okay, that's for taste. And then we'll have a short-finned fish here with spots and tiny barbels. Okay, that's our F1. So now we have 3 genes instead of our 2. And we're going to skip right to the test cross. So this individual will be our test cross chromosome. And this individual will be heterozygous for both the alleles. So they'll have a dominant B allele, a dominant L allele, and a dominant S allele, all on the pink chromosome, and the recessive version of each on the yellow chromosome. Our test cross, remember, is homozygous recessive for each of the alleles. This is our test cross again. All right. So I'm not going to draw out the blue chromosome anymore. That's just going to fade away into the background. But now we can have recombination events in a couple different ways, right? With a 2-factor cross, we could have a recombination event between L and S, right? And that was pretty much it. Either it happened or it didn't. Now we can have recombination between B and L. We can have recombination between L and S, and in some sense generally between B and S, right? So there's a couple different ways we can think about having recombination event. There can be 2 recombination events, one between B and L and one between L and S. So if I just draw out the different chromosomes, we can end up with something like this. We can have a chromosome that's entirely the pink, or a chromosome that's entirely the yellow, right? So we can have the dominant B, the dominant L, and the dominant S together, or the recessive B, the recessive L, and the recessive S. These are our parentals, and they will be the most common. Remember, recombination is relatively rare, so most of the time you see these specific chromosomes. We'll go ahead and count their progeny in the F2. We have 411 here, and let's say we have 412 of this one. But we can have recombination, and we can have recombination in a couple different ways. We can have recombination between L and S, and that gives us the dominant B, the dominant L, and the recessive S. We can have recombination between the B and the L. So we could have that, something like this. All right, so now we have the dominant B with the recessive L and the recessive S. We could have a single, and then we also have the opposite of those two chromosomes. So we could have the recessive B with the dominant L and S, or the recessive B and L with the dominant S. A lot of different combinations. So we have, in this case, we have recessive B, dominant L, dominant S. Here we have recessive B, recessive L, dominant S, right? Several ways that you can have a chromosome resulting from a single crossover. So these are all going to be single crossovers. And they are going to be at some sort of intermediate frequency. So if we were counting them, maybe we counted 61 of that, we counted 30 of that, we counted 28 of those, and 60 of these specific chromosomes, based solely on their traits, right? So this one would have short ball whorls, short fins, and stripes, for example. Then finally, we have our double recombinants, and if you recall from when we drew it out earlier, the double recombinant usually looks like a banding pattern, so that we would have a mixture. You'd either have yellow in the middle or red in the middle, giving you these alleles: a dominant B, a recessive L, or a dominant S on one chromosome, or a recessive B, a dominant L, and a recessive S. These are our double crossovers, and they are rare. Remember, a single recombination event, relatively uncommon. Having two is even more uncommon. Let's say we only count one of those, and we only count two of these. These are actually so rare, if given a table of data and you happen to be missing some categories, that's probably because they didn't exist, right? You had a 0 here, right? So you wouldn't see them as phenotypic categories necessarily. Keep your eye on that for homework, for example. All right, so the question then is, can we take this cross data and can we determine from the numbers alone, here I've drawn out the chromosome for you, but if you didn't have that, could you determine which gene is in the middle? Okay, so there's two ways to go about doing it. I think probably the most straightforward is to just calculate map distances. So we can calculate map distances in a 3-factor cross. And basically it looks like this. For any pair of genes, we have the number of single crossovers plus the number of double. You always count the doubles here. Crossovers divided by the total. Okay, that'll give you, of course, times 100. That will give you the map distance in a 3-factor cross. So in our example, the distance between B and L is going to be equal to any time that you have the B and L alleles swapped, right? So we currently in our parentals, we have dominant B and dominant L together here, and we have recessive B and recessive L together here. So what we're looking for are cases where you have the dominant B with the recessive L or the recessive B with the dominant L. So in this case, we have dominant B and dominant L together. We don't have to worry about those. But here you have one, right? You have dominant B and recessive L together. So you have 30. Here you have recessive B and dominant L together. There's a recombination event that happened between them. So we have to add those 28. And then you always count the double recombinants. Plus 1 plus 2. Divided by 1005, which is what the total ends up being. We have a map distance of 6.0 between B and L. Right, so we found the parental chromosomes. We looked for cases where we have a different recombinant pattern between B and L, and we added them together. Now, if we were looking at the distance between B and S, we have to do the same thing. We have to go and say, okay, we have dominant B and dominant S together. We have recessive B and recessive S together. So anytime you see a dominant B with a recessive S or a recessive S with a dominant B, you have recombination. So right here, we have dominant B, recessive S, so we have to count those 61. Here we have dominant B, recessive S, we have to count that 30. Here we have recessive B, dominant S, so we have to count that 28. Here we have recessive B, dominant S, we have to count that 60. And then we always count the double recombinants. So we need to divide that by 1005 times 100, and we get 18.1 map units. So hopefully you noticed a difference. The distance between B and L, we only added 2 of the single recombinants together. The distance between B and S, we had to count all of the single recombinants together. So if you're ever doing it, if you ever get this kind of question, which gene's in the middle, if you calculate the map distance between a couple of the genes, and any one of them you happen to count all the things, then in those are the bookends. So B and S are on the outside. Because anytime you have a recombination event, right, anytime you have a recombination event between B and L, it's also between B and S. Anytime you have a recombination event between L and S, it's also between B and S. Right, so they're all recombinants. That's one way of determining which gene is in the middle. Whichever one, you find the bookends by seeing which one has all the possible recombinants. There's another way you could do it. It's a little bit more of a shorthand way. Ends up being just as confusing to people, I think, which is to, here's a tip, pick one parental. Okay, we'll pick this one. And then you can find a double recombinant. Remember, are the really rare ones that differs by one single trait. And one trait only, for example, BLS. And that gene is in the middle, the one that's different. So what we're going for is you basically have in our double recombinants up here, we have, look at that, whoa, we have a banding pattern. The middle gene's the one that's swapped out from the parental. So if you pick a parental, like BLS, this one here, the L is the only one that's different. So that would be one of your recombinants. The other opposite would be true then for this parent. The only thing that's different between that one and that one, which is the double recombinant, is that middle gene. But the most tried and true is just to do the map distance and figure out which ones you end up adding together. And to let us practice that, we have another extra credit question. This will be session ID 746869, 746869, 746869. We have a 3-point cross individual where dominant A, recessive B, recessive C are crossed to recessive A, dominant B, dominant C. The progeny is then test crossed. What is the order of genes A, B, and C on a chromosome? Is it BAC, ABC, or ACB, given the result of this cross? Code is 746869. 746869. Seven four six eight six nine. Should be. It's probably hard to see. It's right at the very top of this. There you go. 746869. We'll do another 30 seconds or so. This will give us time to discuss it a little bit. All right, go ahead and put an answer in. I will call it now. All right. All right, support for C, which is the correct answer, though this is a tricky one, of course. So, your parentals, always, first, I mean, the question will tell you what the parentals are in this case, right? But if you didn't know, the parentals are going to be the ones that are super common. Okay. So we know, for example, that we have A, B, or we have A and B together on a parental, right? And we have C here. So if you were going to calculate the distance between A and B, for example, you would look for any situation where you had capital A, capital B, recessive A, recessive B. Right? That would be a recombination event between A and B, going from something that was a mix of recessive and dominant to something that's homozygous, recessive and dominant for both. So anytime there's a distance between A and B, you are going to add them as recombinants. So here, recessive A and B together, that counts. Here, double recombinants always count. Double recombinants are going to be rare, remember. Here, recessive A and B together, so that would count. Here, double recombinant, that always counts. And then two up there, you have two dominant A's and B's together, right? So in this case, every single one of those single recombinants is a recombinant between A and B. And what that'll tell you is that A and B are on the outside, and C has to be in the middle, right? You could have easily picked A and C, gone through this, and noticed that anytime you have capital A and capital C together, you have a recombinant, but that this one does not qualify, and nor does the top one, right? So those are single recombinants that are not between A and C, according to one of the parentals. So you could rule out also that in that sense. Does that make some sense? No? The other thing you could do is you could pick one of the parentals, say this one here. You have dominant A, recessive B, recessive C together. And then you pick one of the double recombinants, like this one. What's different between the parental and the double recombinant? It's the C. That gives you the banding pattern there. Does that make sense? Now, it technically only works between one parent and one of the double recombinants. It's a little harder if we had picked this one and this one, because now the A is different and the B is different, but the C is the same. So you'd have to have picked the other parent to pair with this double recombinant for that trick to work.
But,
What?
I said, but if you pick the wrong one, it doesn't line up.
Right.
So
That's
you just pick the parent and then find whichever one is one of them.
Yeah, so pick the parent, whichever one you want, find the double recombinant that differs by only one letter, and then find out which letter is different, and that'll tell you the middle gene. The one that's different is the middle, because you remember that banding pattern where you start like pink, yellow, pink, right? That's what you're getting here, and that's what's being revealed by that. All right. Well, anyway, that's all I have. So.